Welcome back. In the previous video, we discussed the various toxic factors used by pathogenic bacteria to modulate their environment and the host. However, simply producing these toxins is not enough. Bacteria have evolved a number of mechanisms to mediate the secretion of various protein factors. We will start with bacterial secretion system. The sex system is found in both gram-positive and gram-negative bacteria like B. subtilis and E. coli. Proteins to be secreted have a signal sequence at the N-terminus, which helps the sex system recognize and transport them. These proteins are exported and fold into their active form once they reach the periplasm in gram-negative bacteria or outside the cell in gram-positive bacteria. Now, the accessory secretory sex system is a specialized version of the SEC pathway found in some bacteria. This system is adapted to transport specific proteins that the general SEC pathway can't handle efficiently. It's found in pathogenic bacteria like Mycobacterium, Listeria, and Streptococcus, where it plays a key role in transporting virulence factors that help the bacteria infect and survive within the host. Next, the co-translational signal recognition particle SRP system transports membrane proteins during their synthesis, especially those with multiple hydrophobic regions. Polytopic membrane proteins have several membrane-spanning segments and are inserted into the membrane as they're made. The ribosome pushes the protein directly into the membrane. Bitopic membrane proteins, however, cross the membrane only once with a single membrane-spanning segment.
Twin arginine transport TAT system transports fully folded proteins across the cytoplasmic membrane, recognizes proteins with a twin arginine RR motif in their signal peptide, essential for proteins that need to be folded before secretion, often because they contain complex cofactors. Found in both gram-positive and gram-negative bacteria, commonly used for enzymes involved in metabolism and environmental adaptation. Because of their outer membrane, gram-negative bacteria have at least 6 different mechanisms for secretion. These secretion systems, termed type 1 through type 6, come into 2 flavors, those that depend on the general sex system for transport from the cytoplasm into periplasm, type 2 and type 5, and those that do not depend, type 1, type 3, type 4, type 6.
Type 2 SEC-dependent secretion system. This type involves in 2-step secretion machinery. First, utilizes SEC or TAT system to transport proteins into the periplasm. Second, once in the periplasm, proteins are then transported through the outer membrane via the T2S's machinery that are related to pilus. Exported proteins are hydrolyzing enzymes or toxins such as cholera toxin. Secretion substrates are delivered to the periplasm by the SEC or TAT systems. The inner membrane complex then aligns with an outer membrane pore, and the pseudopolin extends from the inner membrane, binding secretion substrates at its tip. ATP hydrolysis by a cytoplasmic ATPase drives pilin subunits into the pseudopolis, which then extends, pushing the substrates through the outer membrane and out of the cell. The type 5 secretion system T5Ss, also known as the autotransporter system, operates in 2 main steps. First, the protein being transported across the inner membrane via the sex system. The protein is still unfolded during this step. Once the protein reaches the periplasm, it self-inserts into the outer membrane. The protein has a special autotransporter domain that forms a pore in the outer membrane, allowing the protein to be secreted outside the bacterial cell. The autotransporter protein is delivered into the periplasmic space via the general sex system. Once in the periplasm, the domain of the autotransporter inserts into the outer membrane and forms a pore through which the other domains of the protein traverse. In some autotransporters, autoproteolytic cleavage releases the other domains from the domain. The type 1 secretion system is ABC transporter and is made up of 3 main proteins. An ATP-binding cassette ABC protein located in the inner membrane, which provides energy for transport. An outer membrane channel protein that forms a passage through the outer membrane, and an inner-outer membrane fusion protein that spans the periplasm, linking the inner and outer membrane components. Type 1 is capable of transporting unfolded, very large protein. The model is the mechanism of type 1 secretion system. This consists of 3 proteins, an inner membrane spanning ABC transporter. Second is a periplasmic connecting protein accessory factor D, and an outer membrane spinning pore protein. The majority of gram-negative bacteria utilize the type 3. Examples include bacteria such as Escherichia coli, Salmonella, Shigella, Bordetella, and Pseudomonas. Genes encoding T3Ss are often located within pathogenicity islands, suggesting acquisition through horizontal gene transfer. Structurally, T3Ss resembles an injectosome, directly delivering proteins into host cells, and is thought to have evolved from the flagellar structure. Moreover, this system enables the secretion of effector proteins, which typically contain only a component responsible for enzymatic activity, classifying them as exenzymes with toxic effects. The type 4 consists of 4 main component complexes. First, the ATPase, type 4 coupling protein, plays a key role in powering the system. Second, the inner membrane complex anchors the system to the bacterial cell's inner membrane, providing stability. Third, the outer membrane complex spans the periplasmic space. Finally, the pilus structure extends outward, enabling direct interaction with the target cell for the transfer of NAWR protein. The type 6 secretion system is structurally similar to the T for bacteriophage as it shares a comparable mechanism of contraction and injection. When activated, the type 6 contracts, propelling its inner tube outward from the bacterial cell into the extracellular space. As a result, this inner tube can puncture and inject its contents directly into any nearby target cell, facilitating the delivery of effector molecules. Unlike gram-negative bacteria, gram-positive bacteria lack an outer membrane, making their secretion systems somewhat different. Here are some key secretion systems specific to gram-positive bacteria. Sortus enzymes are unique to gram-positive bacteria and play a role in anchoring specific proteins to the cell wall. Proteins transported by other systems like SEC or TAT. This system is crucial for gram-positive pathogens like Staphylococcus aureus and Streptococcus pyogenes, where surface proteins aid in colonization and evasion of the host immune system. Type 7 are primarily associated with mycobacteria. This system is essential in pathogens like Mycobacterium tuberculosis for secreting virulence factors, including proteins that help avoid host immune responses. Exports proteins across the thick, waxy cell wall found in mycobacteria, which provides an additional barrier to secretion. Cytolysine-mediated translocation KMT is a unique mechanism used by Streptococcus pyogenes to deliver bacterial effector proteins directly into host cells. This process relies on a cytolysine, specifically the toxin streptolysin O-SLO, which is a pore-forming toxin. KMT allows S. pyogenes to bypass some of the complex secretion systems typically required by bacteria to inject proteins into host cells.